Changes to a DNA sequence are called mutations of that sequence, so DNA mutations. And that can have several different possible effects on the organism. If it changes the order of the codons that are produced in the messenger RNA, then it can actually result in the production of a different protein, or a protein that doesn't do what it's supposed to do, or kind of a different allele or form of that gene. But remember, we did say that there was some redundancy in the genetic code where more than one codon will code for a specific amino acid. So if you get lucky with your mutation, it may not make a difference at all, because it may just mutate from one codon into another codon that still codes for the same amino acid. So mutations can cause problems, or they may not make a difference at all. So that's kind of one of those things that's left up to chance or up to God, depending on kind of how you view that. But mutations can be detrimental. Typically, if they do manifest, then they are detrimental, but they could also just not make a difference at all in that gene expression. The other thing we need to discuss is that there are ways to regulate gene expression. So all of the cells in your body contain the entire DNA sequence for everything, for coding for every protein you'd ever need. All of the cells have that, but they don't express all of that all of the time. And so what they do is they only express a small percentage of the genes that you have in the cell. And the genes they express, those particular genes, are what make the cells different and kind of gives them their job to do in your body. So the turning off, turning on of genes, or regulating them even more subtly. So making maybe slightly more of one particular protein and slightly less of another particular protein, or stopping production of a protein, that sort of thing is considered to be regulating gene expression. So people that are lactose intolerant, where they don't make the protein that degrades lactose, that gene is pretty much, they don't have that gene. Other people are able to turn on the gene whenever they ingest dairy products. They turn on that gene, and so they make that particular enzyme that digests lactose. So that's something that can be regulated as long as you have the capability to do it. It's something that can be regulated according to when you need it and when you don't need it. The most commonly used method of gene expression regulation is to regulate transcription. Remember, transcription is when we take the DNA molecule and make it into RNA. So what a lot of organisms do, both prokaryotic and eukaryotic organisms do, is they make it hard to do this first step. They make it difficult to get from DNA to RNA. And prokaryotes and eukaryotes do this kind of differently. A prokaryotic cell is a lot more simple than a eukaryotic cell in general. So what they do is they use what's called a repressor. It's a repressor protein, and it's going to bind to the promoter region on the DNA strand, and it's going to prevent RNA polymerase from binding. So on prokaryotic cells, they've got a repressor that's going to bind to the DNA strand. So you can kind of see in the picture here that they've got an inactive repressor in this first one, so it's not bound. But in this one, the repressor is going to bind, and it's going to prevent that RNA polymerase from attaching to the DNA molecule. So if the RNA polymerase doesn't attach, then it cannot transcribe a messenger RNA from that DNA. What eukaryotic cells do is a little bit more complicated, because they're just more complicated cells in general. What eukaryotic cells do is they have a particular protein complex that are called activators, and they have to bind before the RNA polymerase can bind. So they help RNA polymerase bind. So in this picture, and obviously you don't have to know all these names and numbers and all this, but all of these little shapes, these are all proteins that are activators that have to be present and in the right order and in the right shape and in the right location on the DNA for RNA polymerase to even bind. So if one of these components is missing, then RNA polymerase cannot bind and that gene cannot be transcribed. So there are lots of ways that eukaryotes could kind of prevent RNA polymerase from binding if any of these steps, any of these proteins is missing or not in the right place at the right time, then that gene will not be transcribed. Now, there are some other ways that you can regulate gene expression. Two other kind of common methods would be chromosome condensation. So if you keep the chromosome condensed down, then the RNA polymerase doesn't really have a place to bind to it because the chromosome is so crunched. There just isn't room. So the RNA polymerase won't bind. So chromosome condensation, an example of this is X inactivation, which we've discussed before, how females have two X chromosomes, but only one of those is active at any given time, or only parts of one. And so the chromosome condensation is basically keeping the other X so condensed that it's not used to do genes, to do transcription and translation. Another common way that they can regulate the gene expression is through enzymes called proteases. Proteases are found in the cell and they degrade proteins. So if you have a gene and it's expressed itself and you've made proteins, as long as those proteins are active and they're floating around in the cell, they are doing their job. So once you're done with them, you need to degrade them and get them out of the way. So proteases do that. They go in and break down the proteins and kind of get rid of them. They're almost like the trash collectors of the cell. So once they've done that, that will stop that protein from working and basically control kind of how long that gene has been expressed. The last thing I want to talk about is producing recombinant proteins. So this is kind of how we make transgenic or genetically modified organisms, and it's a generally three-step process, although each of these steps kind of has multiple sub-steps. But the first thing you've got to do is you've got to remove the gene that you care about, the gene of interest, from the specific targeted chromosome. So the example in your textbook is for bovine growth hormone. So according to this step one, what you're doing is you're removing the gene that controls the production of this hormone from the cow DNA strand. And the way this is done is through specific enzymes called restriction enzymes, and they kind of act like molecular scissors, and they go in and snip, 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 and basically remove the gene you care about, and they leave kind of sticky ends on that gene, so the gene wants to combine back up with some more DNA. It wants to, it's almost like a magnetic attraction or just something sticky. It wants to combine back in. So you removed the gene from the specific chromosome, and then step two is you're going to insert that gene into a bacterial plasmid. And the reason we use bacterial plasmids, they are circular strands of DNA that are very easy to modify. And they're also very easy to get into bacterial cells. Bacterial cells transfer plasmids back and forth just normally in nature very easily. So if you can insert a gene into that plasmid, it's very, very simple to get it into a bacterial cell. Once you've inserted the gene into the bacterial plasmid, you can now call that a recombinant plasmid. And so step three is going to be insert the recombinant plasmid into bacterial cells. Once they're inside the bacterial cells, then those cells will express that gene, which in the example of bovine growth hormone, the bacterial cells are going to make that hormone. Once they've made the hormone, you can harvest it from them and purify it and then basically inject it back into the cow. So in this picture you can kind of see these steps. So step one is you remove the gene you care about from the DNA in the cow, and then you piece it or splice it into a bacterial plasmid. Then you put that bacterial plasmid into a large culture of bacteria. You grow it. They produce the protein you care about because they're expressing that gene, and then you can separate out that protein, purify it, and get it back into the cow. GMOs and transgenic organisms, which they're pretty much the same thing, are anything that where basically a gene from one species has been inserted into the genome of another species. So anytime you're transferring genes from one species to another, that will be considered a genetically modified organism or a transgenic organism. As your textbook discusses, there are some benefits to this. It makes plants hardier, so you don't have to use as many pesticides. They might survive horrible or worsening environments better. They might have more nutrition. You might formulate it so they're more nutritious. So there are potentially some good things, but there's also some concerns over whether or not these could adversely affect human health. So this is kind of a topic that y'all went into very good detail on in your discussion boards, so I'm not going to beat that to death, but there are some concerns basically over genetically modified organisms.